For all of you budding scientists, entrepreneurs out there, this is what you have to look forward to. It's really great to be back here. I mean, this is such a great community, and it's fun to have grown all the way from MIT into a commercial company now. So I'm happy to talk to you about what we've been working on and the way we've been leveraging smart reagents to validate clinical biomarkers. So I'm now the CTO. I've been the CTO and co-founder. Also, you may have met our CEO walking around. It's an interesting story. I actually met him here at an IdeaStream 5 years ago. So things can happen. So just to put everything in context, it's been about 10 years since we've sequenced the human genome. Now, everyone thought this was going to be the say-all, end-all to diagnostics. We were going to be able to pinpoint a diagnostic outcome with tremendous accuracy. But the problem is, we haven't really seen any impact in the clinic. So why are these data not being translated into useful information? It turns out, at this point in the world we live in, more data is easy to come by. So on the internet, even with genomics, data is easy to come by. The problem is sorting through all of those data to find the relevant patterns that can be used for a clinical outcome. So as a very specific example of this, I'll talk about microRNAs. So this is a class of RNAs that's been discovered about 20 years ago by Victor Ambrose down the road at UMass. And it's been implicated in most major diseases, including cancer, Alzheimer's, diabetes, and one that has plagued many of us in this room, pregnancy. Not that that's a disease, but it just shows the power of what microRNAs are implicated in. Also, they're extremely stable in the blood, so it's fascinating. They come complex in these little microvesicles or tightly complex with proteins, so they're extremely stable against RNA degradation. So they're very stable in the blood. They're also stable for long-term storage. And for all of these reasons, they're pretty much the ideal biomarker. So if you do a Google patent search, you find 50,000 hits for microRNA biomarkers. But if you look today, there's only one diagnostic test that's actually been reimbursed. The problem is that the proper studies are not being performed to actually push biomarker signatures through validation. So if we look at the whole biomarker development process, it's broken up into several different sections. But I'll classify it along clinical utility, so the number of samples, the number of patients that are affected by it. Also the amount of data, so how many molecules are we detecting along this pipeline. Typically, a study starts off in discovery phase, where you take maybe 10 patients with cancer, 10 healthy donors, and you analyze as deeply as possible. You assess all of the molecules that you can detect. From there, what typically comes out is a candidate signature. So you find, in this case, the microRNAs that are implicated in that cancer, and you take them forward into a clinical trial, where you're going to take these tens of markers and push them through 1,000 different patients just to see statistically, is this a relevant biomarker signature? Once you've done your validation, you can take it through clinical use. The problem is most studies done today stop right here. And one of the major reasons is that there are no technologies available today that can actually allow you to detect 10 to 50 molecules over thousands and thousands of patients. So this is where we come in. This was a project that we worked on in Pat Doyle's lab in the basement of Building 66, where we combined two very different technologies. On one side, we have photolithography that they use to print your Pentium 3 processors for computers. On the other side, we have microfluidics, flowing liquid at a very small scale. The streams will not cross-react with each other. And what you do by combining these two is you generate a very fascinating continuous process for making particles of virtually any shape. So anything you can draw on a piece of paper, we can extrude it into a three-dimensional particle. You can make them with multiple regions. So you see down here we have particles that are being made that have two regions, one that has a complex barcode written on it, and another region that contains a biomolecule probe to detect something in solution. We can make them out of any chemistry. It can be hydrophilic, it can be hydrophobic. We can put proteins, nucleic acids. We can actually embed living cells in the particles. We can coat them with viruses. It's quite flexible. One chemistry I want to focus on, though, is a hydrogel substrate. So anyone who wears soft contact lenses, if you pull it out and play with it, that's basically the material that we're using to do our biomolecule detection. So as a chemical engineer, everyone knows that things come down to thermodynamics. So if you want to increase your performance, your sensitivity, your specificity, you need something that behaves well from a thermodynamic standpoint. So just to prove out this point, we used our hydrogel particles and compared them directly to the state-of-the-art polystyrene bead. We did a simple dilution curve to show that the hydrogel gives you about 75 times better sensitivity, gives you 3 logs extra of dynamic range. And because we're using a bioinert substance, we don't worry about proteins or lipids sticking to the particles. We can actually detect in crude samples. Another great thing with the technology, we have complete control over morphology. And this comes into play when we're thinking about performing an assay on our particles, but then how do you read it out? So what we're showing is that we can actually provide a scalable means to do multiplexing. If you look on the left-hand side where it says Firefly, we have a dot pattern that can provide up to 1 million different codes. In the middle there, we have these striped rods that we can scan on cytometers that give you up to 70 codes. And on the right-hand side, if you don't have either of those, if you just have a microscope, we can literally write the name of the target you're detecting on each particle. So what we've decided to do for our first line of products is focus on existing instrumentation. Flow cytometers are ubiquitous in most labs. You'll see there are about 100,000 flow cytometers worldwide, 30% of which are already in the clinic. So what we did is we designed a particle architecture that can do multiplex detection on any flow cytometer. The way this works, we have these striped rods, and in 2 of the regions of the rods, we contain information that is the barcode. So it tells you which molecule that rod is detecting. And in the middle region, that's where we actually detect that molecule. So if you see on the right-hand side, we have scatter plots that come off the cytometer, which don't look very useful until we put them through our software package. So we have a software package that will take information from the cytometers, kind of regroup the particles for what they are, and tell you how much of every target is present. A nice thing about this, too, is most cytometers are equipped with 96-well plate handlers. So this allows us to do 96 samples at once. Our assays are all very high throughput, and they're perfect for validation. So just to look at this process again and see how Firefly can impact it, through the discovery phase, what we found is that most researchers are really lazy or they're really cheap. So people don't want to pay for the sequencing to send it off to a core lab and have this work done. So most people start from reading 2 or 3 papers and saying, well, we have this signature that we want to investigate. We've developed more of an objective method for doing that. We have an algorithm that uses natural language processing to read all of the abstracts in PubMed and build a candidate signature for you based on actual published data. From there, I've already described how we use our assays to actually do high-throughput validation. And as I mentioned, the great thing about flow cytometers is that they're already being used in the clinic. So to implement our assays in a clinical format is very straightforward. I'm very pleased to announce that we've recently signed a distribution agreement with Millipore. So it's great to develop a product, but it's really great to have someone who is going to market it and sell it for you, especially with a global leader like Millipore. Another great thing is, as a scientist, when you develop a technology, when you start to see other people publish on your technology. So we've been very fortunate to work with the leading clinical microRNA research lab who published a nice paper in PNAS. And as one of our very first use cases, we have been working with Victor Ambrose, who discovered microRNA. And he's an avid user of our technology. We meet with him on a very frequent basis. So now I wanted to take a step back. I mean, we're in a unique situation here in that we are a commercial company. We came through the Deshpande Center. I wanted to kind of highlight the path that I've taken to get from where I was 7 years ago to where I am today. So it all started off in Pat Doyle's lab. Pat and I were just hanging out by the microscope in this picture. And you may think it's dark in the lab because the lights are off, but actually his lab was so deep in the basement, it's like a black hole down there. So somehow, even in the absence of light, we were able to publish some great papers in Nature and Nature Materials and Science. And at some point, Pat leaned over the microscope and said, hey, we should probably look at the Deshpande Center. They're doing some great things. So we approached the Deshpande Center. We went through 2 phases of the funding process. As I mentioned, we met our CEO, who was also a Deshpande grantee, but also had an MBA. From there, we saw some real commercial traction, and we decided to apply for some SBIR grants through the National Cancer Institute, who funded us through 2 phase 1s and a phase 2. When you do that, it's really easy to go to angel investors and say, for every dollar you put in, we're going to match it with non-dilutive funding. So we raised some angel funding. But as many of you who've done this know, it looks more like angel funding. So this is where we've come. So we've leveraged the Cancer Institute and also the angel funding to raise about $7 million to get us all the way through the point of commercialization through a big partner. And so now we're really starting to scale up. We're raising a Series A. We're going to start to push out more and more applications and get more and more partners. And I think this is where it really gets exciting. So with that, I'll say thank you, and I'll be happy to see you at the poster. Thank you.